Good morning students. We are studying chapter applications of biotechnology, and as we have studied that biotechnology has various roles in agriculture, in the field of medicine, and we have talked about the preparation of human insulin by using this biotechnology method. And as we know that biotechnology, it is the technique which deals with the uh, playing or the handling with the microbes and their products and their enzymes. So in now the next topic is gene therapy. Now this term, what it is representing, that what is done in gene therapy, the defected gene is corrected. Means if there is any defect in any of the organism, then by using biotechnology the gene can be corrected, because as you know that gene, it is responsible for producing particular characteristics, and if there are any defects in the gene, then the organism is born or the organism have those defects in the body. So what is done with the help of biotechnology, that defected gene, it is removed from the set of the genes and it is replaced by the correct gene, and in this way the right that protein is produced and the right characters are produced in the body of the organism. So, and you have talked about various hereditary diseases, and these hereditary diseases, they can be cured now because of this biotechnology. And this biotechnology, it has a special we can say field where gene therapy is done, and in that what will be done, defective gene will be corrected. And when it will be corrected, it can be corrected in a child, in the embryonic stage also, and after the birth also. Now what will be done? Genes which are defected, they will be removed and the correct genes will be placed in the set of the genes. Now this correction of genetic defective genes, what will be done? Normal genes will be taken and these normal genes will be introduced in the defective gene, and now it can be done in the individual or in the embryo. And what will happen because of that, that known functional gene, which is of no use, or which we can say that the defective gene, it will be removed. So suppose this is the set of genes and here it is defect in the gene. So what will be done? What is done in the gene therapy? This portion is removed and now the correct set of gene or the correct gene is introduced here, and in this way now the organism with the correct set of the gene will produce the desired characteristics. So first clinical gene therapy, it was given in 1990, and to whom it was given? It was given to a 4-year-old girl, and that girl was suffering with adenosine deaminase deficiency. Means we can say that she was suffering from the disease ADA deficiency. Now what is this? This ADA, it is in actual, it is an enzyme, and because of the deficiency of this enzyme, the immune system of that girl was not working properly. So what was done? The defective gene, it was deleted, and deleted gene, then it was replaced by the correct gene. So this old girl, 4-year-old girl, she was suffering from ADA deficiency, and this ADA, it is an enzyme. It is necessary for the production of immunity in the body. Immunity, it is the ability to fight against diseases. And now how this deficiency has occurred? In actual, that gene responsible for this enzyme, it was missing in the set of genes of that girl, and because of that, because the gene was not present, so the body was not able to produce the enzyme. So now if when it can be cured, in some children this ADA deficiency, it can be cured by bone marrow transplantation, and it can also be treated by enzyme replacement therapy. So what is this bone marrow transplantation? We know that in bone marrow RBCs or the blood cells are formed. So this bone marrow transplantation can be done so that this ADA deficiency can be cured. And the second method is that the enzyme replacement theory. So what is being done in this? Because this gene, defective gene, it is not there, or the gene is not there, or the gene is defected, so ADA is not there. So what is done? The what is the function of that gene? To produce the enzyme. So what is being done? That the ready-made enzyme is provided in the body. So this is known as enzyme replacement theory, in which what will be done? ADA will be given by injection. But in both the cases, either we can say bone marrow transplantation or enzyme therapy. What is the problem? In bone marrow, blood cells are formed, and blood cells are not immortal. You know that RBCs, they can live for 90 days, means for 3 months. Then after 3 months again there is need of this bone marrow transplantation, means the patient has to visit the hospital again and again. Then second, if we talk about the enzyme, if when the enzyme is not working, means when the enzyme that has done its work, then again the patient has to visit the hospital for this enzyme replacement therapy. So in both the cases we can say that they are not completely curative. So what is necessary? That at first step, when we talk about the bone marrow transplantation, what is done? Lymphocytes from the blood of the patients are grown in a culture. Lymphocytes will be taken, WBCs, they will be grown in the laboratory condition. Then a functional ADA cDNA. Now what is the meaning of this? ADA, this is adenosine, it is an enzyme. Now cDNA, what is cDNA? It is the DNA which is recombinant, is then introduced into the lymphocytes. So this a functional ADA cDNA, it will be taken, it will be introduced into the lymphocytes, and then it will be returned to the patient's body. So however, as these cells are not immortal, the patient requires periodic infusion. We are talking about bone marrow transplantation, that what is done in bone marrow, what will be done? Some lymphocytes will be taken, they will be treated with ADA cDNA, and now again it will be returned to the body of the patient, but these will produce the blood cells, but blood cells are not immortal. So the patient, it is that will be in the need of visiting the hospital again and again. But how can we solve this problem permanently? Suppose we have to come to know about the problem in the embryonic stage. That's why the pregnant ladies, they are advised to go to visit the doctor periodically, so that if there are any embryonic defects which can be detected in the embryonic stage, so that they can be treated. So the solution is that gene isolated from marrow cells, bone marrow cells, which are producing ADA, they are introduced in the cells at an early embryonic stages. Means in the embryonic stage, what will be done? That gene which has to produce ADA, it will be given to the embryo, and now what will happen? The cells of the embryo, they will be able to produce this ADA, and when ADA is there, then the immunity system will be working. So how it has become possible? Because of the technique of biotechnology. What is done? That the DNA is isolated from the embryo, then that gene is inserted into the embryo, and then this recombinant DNA, it is provided to the body of embryo, and now as it has recombinant DNA, so it will be able to produce the ADA enzyme, and the body will be able to have good immunity. Now the next role of biotechnology is in the field of medicine only, molecular diagnosis. Means what is molecular diagnosis? See, whenever you are suffering from any of the disease, you have symptoms of that disease, and when the symptoms appear, you visit to the doctor. Doctor studies the signs and symptoms, and then the treatment is given. But see, when the symptoms have appeared in the body, means what has happened? Microorganisms, they have entered into the body and they have multiplied. So at this situation, if the disease is chronic or if the disease is very harmful, what will be there? That if many of the microorganisms have entered or many of the cells have been suffered, so what can happen? That the treatment may not be successful at this stage. So what is necessary? That if some of the diseases which are very harmful, their early diagnosis is very necessary. And how we can early diagnose? because whenever the doctor gives the treatment, first of all he gives the test. He may give the serum test, blood test, or urine test, and means when these results comes in the test, it means the infections have spread a lot. So what can be done to avoid any infectious disease or to diagnose that disease at earlier stage? What is necessary? Early detection of that disease. And how early detection is possible? By studying the causative agents, and by studying the causative agents when they are very minute in number or the infection caused by them is very less. So if we talk about the conventional methods जो है diagnosis is not possible at the early stage. So what is necessary? Recombinant DNA technology. We have talked about polymerase chain reaction, PCR. There are two techniques, PCR and Elisa test, enzyme linked immunosorbent assay. These are the techniques in which we can diagnose the disease at the early stages. So what will be done in polymerase chain reaction? Suppose there is a presence of pathogen is normally suspected. When there are symptoms, we have just talked, how can we come to know that the microorganisms have entered in the body? When there is lab testing of the blood or the urine. But up to this time, the concentration of pathogen has increased a lot. So what is necessary? How can we identify at the lower stage? At very low concentration of bacteria or virus, it can be detected by using this PCR, polymerase chain reaction. What will be done? In polymerase chain reaction, this polymerase chain reaction, its uses, they are used to detect the HIV in suspected AIDS patient, and it is being used to detect mutations in the genes and in suspected cancer patients too. Means we have diagnosed these highly dangerous diseases in the early stages. And how it has become possible? Because of this PCR, polymerase chain reaction. So it is very useful technique, powerful technique to identify many of the genetic disorders and many of the dreadful diseases also. So now let us talk about the technique or how PCR is done. What will be done? A single stranded DNA or RNA will be taken. It will be tagged with the radioactive molecule and it is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. So what will be done? A single DNA or RNA, it will be taken from the patient body. Then hybridization of it will be done and it will produce the copy, because DNA and RNA, they have this double helical structure. Now what will be provided it with? It will be provided with a radioactive molecule, and as this as this helical structure, this is the first strand and it has produced the second strand. In the same way, it is the one strand and it has produced the second strand. Now the clones having the mutated genes will hence not appear on the photographic film, because the probe will not have any complementarity with the mutated gene. Means what is happening? Suppose we have taken a single stranded DNA. Now if there is some mutation, then the second strand formed, it will not be able to form the complementarity with it. Why? Because this is defected. How it has become defected? Because some of the microorganisms have changed it. So there will be no complementarity with the mutated gene. And second technique, it is Elisa. So in PCR, let us recapitulate again about the PCR. What is done? A small amount of DNA or the molecules will be taken. They will be amplified, because suppose we have taken a very little small amount of cellular material, and now in the small amount we may not get the accurate result. So what is done in PCR? That cell, it is multiplied, its amplification is done, and by amplifying or by amplification, what chances have increased? That we will get the accurate result, because we have lot of sample of that. So in the early stage also, the diagnosis of the disease can be done by using polymerase chain reaction. Now the second is ELISA, enzyme linked immunosorbent assay, and it is based on the principle of antigen and antibody interaction. Suppose there some microorganisms have entered into the body, so they are antibodies. So what our body will do, it will produce the antigen. So by this ELISA test, it is based on the principle antigen and antibody interaction. Infection by pathogen can be detected by the presence of antigens. So suppose in our body some microorganisms means the pathogens have entered, they are the antibodies. Now our body will produce antigen, and by using this ELISA test, if our body has those particular antigens for particular disease, it means our body is suffering from that disease. And if those antigens are not in our body, it means we are not suffering from that disease. And in this way, this ELISA test, it gives the result of certain diseases. So I think it is clear to you, if those antigens will be in the body, means our body is suffering, means our body will give positive test, and if those antigens are not in our body will not give the positive test. And in this way, in the laboratories, by using these ELISA test or PCR, many of the dangerous diseases can be detected at the early stages. Now the next application or the role of biotechnology is in producing transgenic animals, means the animals whose these genes or the DNAs have been modified or manipulated. Now these organisms, they are known as transgenic because their DNAs or their genetic composition have been manipulated. And now we have many of the animals like rats, rabbits, pigs, sheep, cows and fish which are transgenic. So what is our motive to produce these transgenic animals? So that the production can be increased more and more, and these animals, they benefit human beings by fulfilling their requirements. So now let us try and explore some of the common reasons that why there is need of these transgenic animals. First is normal physiology and development. These transgenic animals, they can be specifically designed. Suppose we want to study some genes, how those genes are working and how these genes are functioning are affecting the functioning of the body and development. So for that we are in the need of transgenic animals so that we can study complex factors. For example, when we talk about the insulin synthesis, artificial insulin synthesis, for that we should have the thorough knowledge that how naturally it is produced in the body, and for that what is done, these transgenic animals are produced. For example, in our previous class we talked about human insulin, and these human insulin, it was produced artificially. But for that we should have the knowledge that how human insulin is produced in the body, and for that the cells of these animals, they are modified, they are combined with the human DNA, and in this way these cells, they give the accurate information that how these genes or DNA, they are producing a particular character. And if we have thorough knowledge that how these genes or DNAs are working in that animal, so the same thing might be happening in the human body, and in this way we can easily study the physiology, so for the normal physiology and development. Now second is study of disease. So many animals, transgenic animals, they are designed to understand that how genes contribute for the development of a particular disease, and for that they are specially made to serve as model for human diseases. So what will be done? DNA from the human body, it will be taken, it will be introduced into the body of that animal. Now the body of that animal or the cells of that animal, they will behave like the cells of the human being, and in this way we can have a study or research on these animals whose cells have been modified by the human DNA. So they will serve as model for human diseases, and so their investigation and treatment becomes easy. For example, cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's, all these are the diseases, And to study about all these diseases, many transgenic animals have been made, and these transgenic animals, they give us full knowledge that how these diseases are spreading in human body. Next is biological products. Now, transgenic animals, they provide many of the medicines, and these medicines, they are helpful in curing many of the diseases in humans. So medicines, they require to treat certain human beings by using their these biological products given by many of the organisms, but these products are often expensive. So what they are saying, that certain diseases of human beings, they are in the need of biological products, but these products are expensive. So how we can take or make them less expensive? By making transgenic animals, and these transgenic animals, they will provide us these biological products which will be cheaper. And how they can be made? By recombinant DNA technology. For example, introduction of the portion of DNA which codes for a particular product such as human protein. For example, we want to produce a human protein. The name of the protein is alpha 1 antitrypsin. What is the function of this protein? It treats the disease emphysema. So what has been done? This product, it is derived from the transgenic animal, and because at this because at it, as it has been obtained from the transgenic animal, it is cheaper, so it can be used for treatment of the disease. Now next example is phenylketonuria and cystic fibrosis. These are the two diseases, and for these also many attempts are being made so that they can be cured by using the biological products, by using the applications of biotechnology. Now next is in 1997, first transgenic cow, means whose DNA was modified, and name of the cow is Rosie. And in 1997, this cow, it was made transgenic, and because of that the milk produced by this cow, it was containing human protein. And because this milk contains human protein, name of the protein is alpha lactalbumin, and it is nutritionally balanced for human babies than natural cow milk, because we know that some of the infants, they are not able to get the milk from their mother, so and they are in the need of that milk because that provides the good immunity system that is necessary for the development of the baby. So what is done, this transgenic cow, these are made so that they can provide the milk which has human protein. Then vaccine safety. Suppose some vaccines have been developed for human cure and they cannot be directly applied on the human body. So what is done, these animals, they are made transgenic. For example, transgenic mice, etc. Because mice, they are representing mammals. So what is being done, whenever the vaccines are made, first the trials are done on these transgenic animals, and if they are successful and reliable, then they can be replaced by the human beings or for other higher mammals. Then chemical safety testing. Now as whenever these medicines or the chemicals are developed, so they cannot be directly applied on the human body, so the testing is done that whether they are toxic or not toxic. For that also transgenic animals are made, and from these it is find that if they contain any toxic substances, and then they are exposed to toxic substances and their studies are made. So toxicity testing in such animals will allow us to obtain results in less time. So these are the certain reasons for which there is need of these transgenic animals. But now as this biotechnology, in this we are using many of the microorganisms or plants or the animals for the benefit of human beings. So it has some ethical issues or the moral issues also. So this manipulation of, because what we are doing, we are manipulating the living organisms. How we are manipulating? We are changing their DNAs, and if their DNAs are changed, so what is being done? Manipulation. And why it is done? For the human race. So what should be there? This earth, this planet earth, it has it has given equal rights to all the living organisms present on the earth. So there is a regulation of this genetic manipulation. For that, some ethical standards are raised which determine or which evaluate the morality of all human activities, that whether these activities are harming or not to the other living organisms. Or going beyond the morality of such issues, suppose we are making recombinant DNA, or we are making them genetically modified, means what we are doing, we are disturbing the genetic composition of that organism. And this genetic modification of the organism, it can lead to unpredictable results, means there may be certain results which can be more harmful for the human being. So there should be a control over this. Then next, for that, Indian government has set up an organization, and that organization, it is named as GEAC, Genetic Engineering Approval Committee. And this committee, it takes the decision that whether the genetically modified researches, whether they are in the field of plants or animals, whether they are right or valid or not. And if they are not valid, then GEAC does not permit them to make that genetically modified organism. So this is the control. Then modification of living organisms for public services has also created problems with patents granted for the same. Now what is patent? Patent, we have talked that certain productions, they have their marks which can be produced by those countries, those companies only. So they have taken the patent for that from the government. But when these genetically modified organisms are made, then they have created some problems with the patent also. So what is necessary, that there should be a control. And let us give me one example. Then if we talk about this DDT, when this DDT, it came into existence, it is a poisonous chemical, and it is sprayed on the insects. And when it came into existence and it was sprayed on the insects and mosquitoes, then what happened? In the beginning, many of the insects and the mosquitoes and died. But later on, what happened? Some of them became the resistant to that DDT, and that's why the government, and now as they have become resistant, and this DDT, it is non-biodegradable. Now the insects are also not being killed, and this the DDT, as it is a poisonous chemical, it is entering into the food chain, and it is causing damage to the, or causing certain loss to the human beings also. So what has happened? The organism has become modified, or the organism has brought some genetic modification in its body. In actual, we have not done anything. Biotechnologically, human being has not done anything. But because of this DDT, the genetic modification of the organism took itself. अपने आप evolution हो गया, and because of that, this DDT, it is causing some side effects or loss to the human beings, and the main motive was to kill the insects. That is not taking place. Now there is public anger also, that certain companies, what are they are doing, they are taking their using their patents, and they are using the products and the technologies, and then they are selling those products by putting their tag marks. For example, if we talk about the neem or the tulsi or the aloe vera, all these are mentioned. Uses of all these plants are mentioned in our Ayurveda also, means India has the patent for all these products. But what the other countries are doing, they are using this, and now they are giving their names, they are putting their labels, the companies are putting their labels, and they are selling the things in the market. So that's why the local people, they are also becoming angry in this. Now rice, it is the important food grain, and in India also it is produced in large amount. And if we talk about the varieties of India, varieties of rice, near about 2 lakh varieties of rice are grown in India. And if we talk about the diversity of this rice, we can say that India has the richest diversity in the field of rice. Basmati, it has its unique aroma and flavor. And if we talk about our literature, our previous history, then Indian history, it is mentioned about the 27 varieties of the basmati. And these from where we have taken all these records, these are mentioned in our ancient text also, folklore also, and poetry also. But in 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. In actual, this variety, all these varieties of basmati, they are grown in India. But what that company did, उसने अपना trademark लगाया and he took the patent. That company took the patent from the government of America, and in this way this allowed the company to sell a new variety of basmati in the US and abroad. Now in actual, this basmati, it is the derivative or the product of India. But what this company did, that the label or the trademark of that company, it was taken granted from the government. And how this was made, Indian basmati, it was crossed with dwarf varieties, semi dwarf varieties, and then they said that we have invented a new crop. So Indian basmati, use of Indian basmati was done. It was crossed with semi dwarf varieties, and that country, what they did, they gave a new name of basmati, and then they took the patent. Now implying that other people selling basmati rice could be restricted by the patent. Now what is the loss of that? Because of that, that company got the patent. Now that company can only sell those rice. Now where it was grown earlier, for example, if it is grown in India, the people of India cannot sell that rice because that company has taken the patent. Only that company can only sell the rice. So these are the reasons of anger which are causing great loss. Now biopiracy. Biopiracy, it refers to the use of bioresources by multinational companies and other organizations without authorization from the countries. Just we have talked that what the multinational company of America did, that took the basmati rice and crossed with the semi dwarf varieties, and then they gave their name. So using this type of activities or doing this type of activities, this is known as biopiracy, and without any compensatory payment, means they are not paid anything for taking those species and using them as their name. This is known as biopiracy. So most of the industrialized nations are rich financially. If we talk about India, India is not very developed, but it has lot of biodiversity. It has different varieties of floras and faunas, and it is rich in the diversity. And because of that, many of the strains or the varieties of India's are exploited, just about which we talked, that how the rice basmati has been manipulated, or the use of aloe vera or tulsi has been done by the other countries. So what should be done, and what is happening, that there has been growing realization of the injustice. Inadequate compensation means injustice is happening, they are not getting any compensation or amount also for that, and between the developed and the developing countries. And some nations are developing, and that's why some laws have been laid so that the exploitation of these bioresources may not take place. In Indian Parliament also some amendments have been done in the Indian Patent Bill. And what are these amendments? That these points are considered, in which the main strains or the whatever the products of the India are, they should given the name of the Indian product only. And so this is about how the various laws have been made, and this is all about the chapter biotechnology and its applications.